All right, so in this series of lectures, we'd like to talk about DNA replication, the duplication of genetic material. And as we mentioned before, no matter what the genetic material is, whatever it turns out to be, in our case it's DNA, whatever it is, it has to, one of the properties is that it must be able to replicate with high fidelity. So it must be able to be reproduced, and that due with a minimum of error rate. So why would we do this? Well, it's pretty simple for many reasons, but first is we need to have cell division, right? So replication, while it's not physically part of cell division, it is the precursor for cell division. Before you have effective cell division, you need to have replication. And it's fairly easy to think why. If you go along and you don't replicate your DNA first and you simply split it apart each time, you get one half each time. So you always want to make sure that when you create a new cell, that it has the same genetic information as the original cell, except in the case, of course, of meiotic cells or gametes, where you want to cut it in half. But even there, you still have to replicate it. Now, in general, why do you need to go through this whole process? Well, one simple answer is just a matter of cell surface size. If you think about surface area to volume, if you have a small volume, you have certain surface area to that. If you increase that volume, you have a very large change in the ratio, a reduction in this case, of the ratio of the surface area. What that means is when you increase the volume, you have less surface area per unit volume. In a small cell, you can easily transport oxygen back and forth. You can transport materials that you want to remove from the cell, waste materials in and out. Once that structure becomes too large, it becomes physically impossible, or intractable at least, to actually effectively move material out. And you've seen these old movies from the 1960s and 50s where there's a huge blob thing that was a single cell that was the size of a building. This simply can't happen because you couldn't, there's no infrastructure to hold it up, but besides that, even if you couldn't move materials in and out that it needed in order for it to respire and go through its normal processes. So one's just simply cell size. The other, of course, even when you don't worry about size, is you need to replace tissue. A good example would be skin cells. Our skin cells are constantly sloughing off. In order for that to happen, you have to have meiotic division going on in the basal cells. You have differentiation into cells that are going to function as these basic cells, skin cells, and then of course you shed them. This same thing happens in our intestine each time we ingest food. Part of our intestinal wall goes away with that food, and so they're underneath, they're meiotic cells. These produce cuboidal cells, which then form the lining of the intestine as well as the lungs. So tissue replacement is an important thing to have. Okay? Of course, sexual reproduction. In order to have the proper amount of DNA in each of our gametes, even though we're splitting it down, in order to produce the right complement, we first have to have replication go on. So long before we start the process of making gametes, we have to actually replicate. And then once, of course, we fuse those gametes into an embryo, we have embryogenesis. Embryogenesis is simply just the duplicating of cells and differentiating of the cells through mitosis. And the cells start out single cell stage, they divide up into blastulas and all sorts of things, eventually producing new, through the rules of epigenesis, producing new eyes, new livers, new hearts and things. All of this done by the process of mitosis. In order for that process to occur, you have to have replication occur first. All right. So let's talk about DNA replication. Talk about this fairly complex problem that we are only, after 50 years, still trying to understand. Okay. Now, just to point out what's going on here, we take something like an E. coli. Okay. E. coli has approximately 5 million base pairs in a single chromosome, and it takes less than an hour for that replication to occur. A human cell has 6 billion. Remember I told you that there's enough DNA in a human cell to stretch from here to the moon if we were to take all of our DNA and literally stretch it out. Still, all those billions of base pairs of cells can be copied in a few hours, and they are just phenomenal how that can work, okay? And what's really, really important is that the accuracy of this is just absolutely phenomenal. Okay, the high fidelity truly does hold. We see that the process has one error in every few billion nucleotides. Now, there are lots of errors. What happens is you don't end up with those errors because we have a proofreading system. Just like when you're typing on your computer and you misspell the, T-E-H, and it flips the E around, You have the same thing going on in your DNA, fortunately, but during replication there's a proofreading, and we'll talk about that. Okay? All right, now this next slide is very important. This slide is important not because of just the information on it, but because we really need to put something into your brain that just doesn't seem to be there for a lot of us. That is the timing of when replication occurs. If I ask the question, when does replication occur? One of the most common answers I get is somewhere here in prophase. Replication occurs in prophase or right before prophase. And it's not true, okay? Mitosis and meiosis have absolutely nothing to do with DNA replication in the sense that it's required in order for that to happen. It's a precursor, but it can be hours, days, in fact it can be years in between S phase, which is replication or synthesis, and the actual mitosis or meiosis problem. Now for mitosis it's not years, okay, it's very short, but certainly meiosis, if you think about human females as they reach puberty and adulthood, they start to have ovulation about a monthly cycle. Well, those cells that are being ovulated have been arrested up here in meiosis. The replication that occurred for those cells actually occurred right before birth. So if you're a 25-year-old female, that's been 25 years between S phase and the end of your meiosis phase when you ovulate, complete the oogenesis and ovulate that egg. So this G2 phase is pretty important. So it's really important. People just seem to, and I think it's because we call all of this interphase down here. And it's like some phase that's part of mitosis. It's not. Interphase is the life of the cell. And mitosis or meiosis is a completely different time. And we'll talk more about that when we cover meiosis and mitosis. All right. Mitosis, of course, just to remind you, is where we're going to divide the DNA up and get two daughter cells that are essentially identical to the cell we started with. They're just smaller. Meiosis, on the other hand, we're going to keep dividing so that we get haploid cells. We cut the DNA concentration into half in each cell. And that'll be important when we talk about Mendel and other things next. Now, just to show you that this is not something new, obviously, even though we still don't understand it very well, when Watson and Crick put out their famous paper outlining the structure of DNA, they actually mentioned that it was possible that there was this mechanism, that the design that they had for this double helix lent itself to a certain type of mechanism of replication. Okay? And they said, basically, what they did was a few months later, they published another paper outlining how they thought replication occurred. And they said, what we see here is this. We see this process where the DNA helix is unwound during replication. The unwinding leaves us with one strand that we can copy over here. That becomes a parental strand with a new strand connected to it. Okay? And then we have another one on the other side. It works because of complementarity. If you pull it apart and you have A, G, C over here, then you're going to put a T, a C, and a G down here. So you're going to have the complement being built. All right? This is known as semiconservative replication. All right? Semiconservative. Only half the original strand remains, and the other half of that strand becomes a completely new piece of DNA. Now, why would we worry about that? Well, for several reasons. One, there were multiple models at the time about how replication might occur. Watson and Crick proposed what was known as semiconservative. At the same time, there was one called conservative, and there was another model known as dispersive. Now, dispersive is a little odd, okay? It basically says that we're going to cleave everything up. I'll explain why we thought that was a possibility in a moment. But Watson and Crick said, no, what we're going to do is pull them apart, and we're going to make copies of each side, and there's going to be new strand and old strand. Okay? And this was kind of opposed to the other models. The first model being conservative, which is that you would pull your DNA apart, okay, break those hydrogen bonds, pull it apart, somehow make copies, and then somehow those new copies would fold around, and the old part would come back together. So that you would always have the original double strands here in blue, and you would make the red being the new copies. Under dispersive, you somehow cut up pieces of the DNA so that this piece up here is made new and this is old, and I'll show you why people didn't understand how that could happen, but some of the experimental data actually suggested it might at first. And then on the Watson-Crick model, like I said, you pull it apart, you end up with a new strand and an old strand, new strand and old strand, new strand and old strand as you work down. So you're always gaining a new version of your strands. Okay? All right. So how do we know that it was semi-conservative? It was proposed, but how would we figure it out? Well, there were a series of experiments, and your book talks about a couple of them. I'm going to highlight Taylor, Woods, and Hughes' experiment in 1957, where they demonstrated that semi-conservative replication occurs in eukaryotes. Okay? They use fava beans. They use the root tips because root tips of plants grow really, really quickly, and they do it by mitosis. And so what they basically did was they put heavy thymine, so radioactive hydrogen H3 thymidine in. They let it incorporate into the chromosomes during replication, and they exposed it to X-ray film, as we see here, and they came up and showed that, yes, they've got all these new patterns. Now, what they then did was let the replication go a second time without adding any radioactivity. So the idea being that you've got these radioactive strands, you pull them apart. If it's conservative replication, all your radioactivity will end up back together, and then your new strand will show no radioactivity. If it's semi-conservative, you're going to have partially put together. And so they allowed this to occur on. They went through the process. They separated it out, and lo and behold, this is what they got. Okay, you'll notice that this is a really good one right here. That's the centromere. So this is a real short little chromosome with this. It's upside down. Right where my red dot is the centromere. And one arm is hot. That's the little beads you see on it from radioactivity. The other arm is not hot. So this was semi-conservative because remember, we're at the second round right here, okay? So if we go back to our picture earlier, you think about it at the second round, you shouldn't have anything. And the same thing is happening up here, okay? So this was one of the definitive experiments. There were several again, but this is one of the definitive experiments that showed that semi-conservative replication worked. Now, back to dispersive. Why did anybody think of dispersive? Well, there's a good example right here. If we look at this big chromosome right here, notice that there's very little or no radioactivity over here. Radioactivity is limited on this side over here, but every now and then there's a little bit of radioactivity, like right there. Well, what that was, they're going, how could that happen? We're in mitosis now, remember, not meiosis, so there's no recombination supposed to be there. Well, lo and behold, there does. What happens is, even in mitosis, when you don't have this formation of these big tetrads, you just have sister chromatids, there still can be some crossover. Okay? You still get some exchange, and that just demonstrates that exchange. So when people had seen these experiments early on, they thought it was dispersive. They thought there was a piece of new DNA and a piece of old DNA, when in fact all that was happening was that things were recombining a little bit. No, it's not recombination, it's actually just crossover. Okay? So that's where that is. That's why that exists. But this is the good one right here. This one right here shows you that semi-conservative was the correct model, and that's the way that it worked. Okay? So where are we now? We have three different mechanisms proposed for replication. Semi-conservative, conservative, dispersive. All of the data, and even to this day, points to this one model over here, the semi-conservative model. We start out with double-stranded DNA. We open it up. We make a new strand on each side so that we go back to being double-stranded, and at the end we become old and new. Okay? So somewhere out there, we have old and new. So all those parts are listed. Okay. All right. So that's where we get to on this. In the next lecture, what we're going to move into is actually talking about DNA replication and how all this occurs. All right? So that'll be now. So for right now, we've just talked about how we got to the basic model, the big picture. Next, we'll talk about the specifics of the actual model itself.